So we'll just get started and people will be joining us as we get going, but I want to welcome everybody to kind of 101 Cyanobacteria with Karen Seaver. I'm Susie Spickle, the Community Programs Director at the Harris Center. I'm one of the naturalists here. And before I introduce Karen, which will be my pleasure, I just did my intro. Great. So now I get this pleasure of introducing you all to Karen Seaver, who is our staff ecologist here at the Harris Center. Karen also teaches at Keene State College, and I've seen Karen in action teaching. She's a wonderful, wonderful educator, a fabulous scientist, and it's an absolute pleasure to have her on staff here at the Harris Center. She spent time in Antarctica. Her specialty is soils, and with the Harris Center, she's really taken on a big cyanobacteria project that I'm sure she'll be talking about, but it's really exciting. It's kind of cutting edge, a new way for citizens and community members to be involved in monitoring our ponds and lakes for cyanobacteria. So Karen, welcome and thank you.
Thanks so much, Susie. And thanks to you all for being here. Oh, tonight you're here, hopefully you like water, and you're gonna hear about aquatic systems a bit. So tonight we're gonna talk about what you need to know about cyanobacteria. Oops, I gotta be able to advance. So here's a little outline for tonight. I'm going to try to put cyanobacteria in a bit of evolutionary and ecological context to give you sort of a window into their world and how they impact our world. And then we'll move on to some of the current and really, like Susie mentioned, really front happenings in the cyanobacteria monitoring and some of the trends that are happening. I'll talk a bit about local monitoring in New Hampshire and what the Harris Center is doing on local lakes, especially Norway Pond here in Hancock. And then I hope to give you some takeaways about things that you can do to help water quality in your area. And then, like Susie mentioned, I'm happy to take questions. Susie can feed them to me throughout, so you can put them in the chat or you can save them for the end. I will try to leave a nice chunk of time at the end to talk about whatever you're interested in. So a bit about cyanos. They're hard to categorize. One of the complicated things about cyanobacteria is they have a lot of morphological and functional overlap with the true algae. And in the past, and still by some folks, cyanobacteria have been called blue-green algae. And the blue-green algae fits partially. The blue-green part works just fine. But interestingly, they're not true algae because of their cellular structure. So if you look on the right side here, you can see two basic types of cells. Prokaryotic cells lack a nucleus, which is a membrane that surrounds their genetic material. And eukaryotic cells, we're eukaryotes, our cells have a nucleus. Algae are eukaryotes, and cyanobacteria are actually bacteria, meaning they're prokaryotes. They're pretty, in some ways, quite primitive organisms. And in fact, as far as we can tell from the fossil record, cyanobacteria are some of the first organisms on our planet to evolve on our ancient planet. And when they got their start on planet Earth, they were chemosynthetic, meaning they didn't perform photosynthesis. And we know that because there was an absence of oxygen in the atmosphere of early Earth. We know from looking at the geologic record that photosynthesis didn't evolve until about 2 billion years ago, but cyanobacteria fossils date much earlier than that, and I'll talk about that in a minute. Cyanobacteria are everywhere, and they've always been there. We've only given them sort of more recent attention because our tools in order to be able to monitor them have gotten better. And also we better appreciate some of the health and environmental concerns that go along with that, this group of organisms. So they are most commonly found in aquatic systems, freshwater and saltwater systems, but you can find them in terrestrial environments, and you can even find them inside rocks. That's we call those cryptoendolithic rocks. And in specialized environments such as Arctic and Antarctic systems, you can find these cryptoendolithic structures, which are really quite amazing to think about how these organisms survive. So cyanobacteria are not invasive. They're native to our local lakes and ponds, and they're typically found at really low abundances, and therefore we can't see them, but they're there. So the next time you go swimming in a freshwater body or a saltwater body, it's just teeming with microscopic life, to the point where you get out of the water after your swim. Yes, you're dripping water, but you're also literally dripping organisms. And some of those organisms are algae, true algae, and some of them are cyanobacteria. Some species of cyanobacteria produce toxins, and we call them cyanotoxins. But what complicates the picture further is not all species of cyanobacteria produce toxins, and even a known species that's known to produce toxins, it's very strain specific. So not all strains of that species will be toxin producers. So it becomes a challenge to really learn what's happening in this system. So at the top right here that's bumped in, this is a picture of a local cyanobacteria colony from Norway Pond. A microcystis colony, and microcystis is known to produce toxins, and it's found in our local lakes. But what's interesting is that it's typically found at levels that are not going to be harmful. But what we need to keep our eyes out for are when we have an overabundance of these organisms, and I'll talk about that in a bit. So some evolutionary context. This is what's called a phylogenetic tree. And I just want to touch on this to really show how the lineages of cyanobacteria and true algae are, they're sort of evolutionary cousins, but not brothers and sisters. So if you're looking for cyanobacteria, it's over here in the prokaryote domain, one of the two prokaryote domains, bacteria and archaea. And then over here, you have the eukaryotes, organisms that have a nucleus. If you're looking for your own species, here's the animals. And then the algae are over here. Algae, true algae and land plants are much more closely related than algae and cyanobacteria. So it's like compelling, right? How organisms could be have very distinct evolutionary lineages, but have such similar functions and morphological appearances, at least at the levels that our eyes can detect. So while we're talking trees, this is just a note to say that algae taxonomy is complex. I mean, taxonomy is always complex, but while we're talking trees, different kind of trees than most of us are used to looking at, this is sort of a more detailed phylogenetic tree that sort of shows some of these interrelationships. There's some other sort of organisms in here that you might recognize as being part of like the algae group, meaning organisms like dinoflagellates, diatoms, chrysophytes. These are organisms that are autotrophic and they're green, but we have to dig a little deeper in order to really understand sort of truly who they are. So we're just really starting to get a handle on diversity of this group of taxa, and modeling estimates put the number of species of cyanos at about 6,000, which for a relative scale, that's about as many species of mammals as we know of here on our planet. And eukaryotic algae, it may be because we've been studying them longer, but we estimate their diversity to be a great bit higher at maybe close to 40,000 different species. So teasing them apart requires a combination of microscopy and also, genetic analysis using, DNA sequencing. Okay, so back to some more familiar bacteria. I mentioned earlier about how cyanos are some of the first organisms to evolve on our planet. And what's also kind of mind-blowing is that many of their descendants are still alive on our planet. So the picture on the right here is showing a fossil stromatolite. Stromatolites are types of cyanobacteria. And this one is found at Glacier National Park in Montana, because you may know that the central part of North America used to be an ocean in a few millennia ago, or a few billion years ago. And then the picture on the left is a living stromatolite. It doesn't look like an organism. It looks like a rock or a crazy sort of piece of concrete, but that's a layered, structured group of organisms, and they sort of kind of like coral grow from the bottom up. So the more recent strata are the sort of living strata here at the top, and the ones sort of closer to the bottom have died off, and they're just sort of providing structural support for the organisms at the top. So most stromatolites, not all, are marine. So that's why you can still see them. This one's the most famous group of stromatolites is from Shark's Bay in Western Oz. So definitely a place I'd love to go someday. So the earliest fossils we have of stromatolites are 3.5 billion years. And if you follow along with what astronomers think the Big Bang happened, that was just a bit more than 4 billion years ago. So it's amazing that these organisms evolved and started to colonize Earth to the point that they could form fossil evidence by just a bit more than 500 million years. Amazing. They're true extremophile survivors. And then, as they went through on their evolution, we have to thank them and other groups of organisms, including the true algae, for their role in the oxygenation of the atmosphere, without which we heterotrophs would not be here. So kind of an unsung hero of our planet. How about cyanobacteria as food? Maybe, depends on the. That's a form of cyanobacteria that has lots of health benefits and is also really tasty. So the picture on the right here is 2 different types of spirulina cyanobacteria under the microscope. And on the left is what they typically look like in a spirulina bowl, which is quite tasty. It's sort of, to separate a bit of mind, your eyes and your stomach, but so some strains of cyanobacteria are consumed by humans, and in the wider ecosystem, cyanobacteria are consumed by all manner of organisms in the ecosystem. So they have a really profound ecological role, especially because they're primary producers. And as such, they help support the trophic systems, not only of aquatic systems, but also of terrestrial systems. So you can see by this trophic diagram, there's aquatic organisms represented, and there's also terrestrial organisms, sort of showing that this group of organisms is highly influential. Of course, these organisms are performing photosynthesis, so therefore they're involved in climate regulation on our planet. They're helping to sequester carbon dioxide out of the atmosphere. But the turnover rates can be pretty huge. And that's what we'll talk about a bit later when we talk about what happens when blooms occur. So this tight-knit ecosystem and community structure is, part of what makes nature work. But it also poses concerns when you think about toxin production. because these toxins persist in the environment. They do not degrade easily. So they have this habit of undergoing what we call a two-step process: bioaccumulation and then biomagnification. So bioaccumulation is sort of the idea that you are what you eat. If you eat something that has a persistent toxin, that persistent toxin then becomes part of your body. And if something eats you, it gets passed along. And the higher trophic levels, the higher level predators, they experience not only bioaccumulation, but biomagnification, where the prevalence of the toxin in body tissues, the concentration of the toxin, magnifies by at least 10-fold with every trophic level. So there can be significant effects, especially at the highest trophic levels. And as humans, as the top of the proverbial food web in many systems, we of course need to be mindful of this too. It also, of course, affects wild species and has significant ability to affect our livestock and pets. And I'll talk about that in a bit. Okay, so blooms. Blooms are what you came here to hear about, I bet. So lots of different things can bloom. What does a bloom mean? A bloom just means there's enough of it that you can see it, that the levels of the population abundance have become high enough that they're visually apparent. They've been there all along. But when the blooms form, there's a really incredible diversity of their physical presentations. You can have scums, you can have films, you can have blobs suspended in the water, you can have filaments, just general water discolorations or cloudiness. Sometimes, but not always, there's an odor associated with the blooms that kind of smells like, rotting plants. And if you have cyanobacteria present that are producing cyanotoxins, they will increase in concentration along with the bloom. And there is where we enter into significant ecosystem consequences and consequences to organism health. One question that people often ask is, what can we do about blooms? The treatment is difficult. It's challenging at best, if not impossible. There's a couple physical methods of control that have been tried, things like aerating the water. There have been some chemical methods that have been utilized, such as silver sulfate treatment, some other, trying to get sort of sedimentation to occur where the bloom sort of breaks apart and sort of settles to the benthic part of the system. But all those approaches don't always work. They're often expensive and hard to deploy in a lot of areas. And the trick with blooms is they can come and go really fast. So it can be difficult to even deploy some of these strategies. So it's important to remember that these organisms were there all along. They just become present at a level where the microscopic now becomes macroscopic. And on some level, it's an alert. It's a sign that we need to be watching for what's happening. And in some level, it's an indicator, right? That there could be other water quality issues that are occurring that we need to think about what we can do. So we know in cyanobacteria are increasing in frequency and severity, and they are increasing due to many factors. This is, another part of the threads that are complex. So one thing that's in the caption of this figure that you might see a lot, an abbreviation, HAB, means harmful algal blooms. And it's an acronym that's starting to be phased out because we're trying to call them what they are, which is cyanobacteria and not algae, but it's a moniker that still persists. So if you ever see HABs, that's harmful algal blooms. So this figure on the left, this column chart, shows how we first sort of became really aware of cyanos in the 1960s. And in fact, that's when they were first described here in New Hampshire lakes. And since then, with every passing decade, there's been a marked increase in not only the number of incidences of cyanobacteria blooms, but also the number of publications and studies that are being done on this group of organisms, which is great because there's a lot to know, and we're only going to get better with sort of understanding the system and try to tease apart all these factors. So let's talk about some of those factors. Weather affects blooms. You can have something as simple as high levels of rain followed by high temperatures. That can result in a bloom activity. So with a high prevalence of precipitation that we've been experiencing, at least here in New Hampshire over the past month, it's been a bit of a blessing that we haven't had super high temperatures following those rain events. Otherwise, we would see probably even more blooms than we have so far this summer. The reason why the rainfall matters is the thinking is at least that it flushes nutrients from the terrestrial environment into the aquatic environment, and that sort of primes the system. And when that temperature rise occurs, sort of the factors align and you see that bloom activity take place. If water is relatively still or even stagnant, you definitely have a higher tendency for blooms. And there's a really big tie-in with a lot of the factors that cause another ecological process that you might have heard of, the process of eutrophication, where water bodies become increasing in the amounts of nutrients that are found there, especially nitrogen and phosphorus, but other micronutrients as well. These often come from a variety of human sources. Agriculture, especially fertilizer applications, and livestock waste. When that leaches into our aquatic systems, that provides a lot of the raw materials that are really kind of craved by a lot of cyanobacteria. If you have industrial outflows of waste products that are nutrient rich into systems, or even thermal pollution where the water is entering a water body at a higher temperature, that can influence bloom rates. And then one that ties in with a lot of our lake lives is sewage leaks. Sewage leaks from folks that have septic systems that are proximal to water bodies. Even if you only have a tiny leak in your septic or an overflowing leach field, that can cause a really large nutrient pulse. And of course, climate change, right? Global warming, increased average Earth temperature is laying the groundwork for increased bloom frequency, and also persisting far later into the months. We're even seeing blooms happen in the fall and even winter. There's been reports of blooms happening underneath ice lids in lakes. So the game is really changing because of the changing climate. And the microbes can respond really fast. They're short-lived, rapidly turning over organisms that can, more quickly adapt and take advantage of these changing systems than long-lived animals can. So there's a lot we still need to figure out in terms of how, climate change, and, climate change isn't just temperature, it's also affecting precipitation shifts. So there's a whole ball of wax that is involved here, but it's important to remember that there's some of these environmental conditions that predicate blooms that are beyond our direct control. We can't stop the rain, but there are some, and we'll talk about this at the end of the talk, things that you can do, things that are within your control that can not stop blooms from happening, but maybe turn down the volume on some of these factors that are then sort of laying the groundwork for them to occur. So it's always encouraging, I think, to sort of, take action. And I'll talk about that in a little bit. Okay, so now we're going to talk a bit about, so what, who cares, right? you know, if you care about the environment, then I think we made a pretty strong case. But if you're the type of person who's more human-centric, and there's a lot of us out there, let's try to talk about some of the human health concerns associated with cyanobacteria. So here's a list of common cyanobacteria genera. That's typically as far as we go in terms of classifying them by the microscope. They have all these amazing names that are also difficult to pronounce. And they come in incredibly diverse forms in terms of how they look at the surface of the water and under the microscope, and then their association with known toxins. So let's just look at the one at the top of the pops here. It's a cyanobacteria you might have heard about. And for many, many decades, they've been called Anabina, and they're starting to be reclassified. It's the same genera, but we're starting to call them Dolichospermum. I don't know why, ask the taxonomist. But look at how many different toxins this one genera produces. They're hard to categorize. And then these different toxins, in general, cyanobacteria produce predominantly neurotoxins and hepatotoxins. So toxins that affect our nervous system and toxins that affect our digestive system. So they have been implicated in a number of different human health concerns. And if we look, let me make sure, I don't know if you see this, I gotta make sure I can share my screen. I'm trying to show you some of the pictures of them. And if you're curious about resources, I'm going to show some of them at the end, but if you are someone who's thinking about getting into studying cyanobacteria in your area, there's so many great resources out there. And one of the best ones out there is this one, and it's by the UNH Center for Freshwater Biology. And you got to love the name, the Dirty Dozen, the Toxic Cyanos of New England. And here, shout out to Amanda McQuaid from New Hampshire DES, who did this as part of her graduate work. You can kind of go down the rabbit hole of looking at the different descriptions and forms of these organisms. And most of these are microscope pictures, not sort of pictures of the blooms at sort of water level. So it's amazing to see how different they all can look for all sort of being somewhat similar. So some of the ones I'm going to talk about later, I'm going to talk about Anabina Dolichospermum, because that's bacteria that was noted in a bloom at New Benusset Lake last November. And later I'm going to talk about Oscillatoria, or the Oscillatorales, and this was the genera that was implicated in the bloom at Norway Pond last summer. So these are around. We don't necessarily have all of these cyanos in every lake, but you can find these quite commonly throughout New England. Okay, so how do we get exposed as humans and our pets and livestock? The four main ways of exposure are through our skin, through breathing, through swallowing contaminated water, and eating contaminated food. Probably a lot of these aren't that surprising to you, maybe other than breathing toxins. Cyanobacteria are small enough that they can aerosolize. So if you're sitting by a pond, looking out at the lovely vista and breathing in the humid air, you're also breathing in cyanobacteria if they're there. And if they're occurring at really high levels, that can whip enough of them into the air that they could, in theory, affect your health. You have to be a little bit more concerned about the last two on this list, swallowing contaminated water and eating contaminated food, because those are the two pathways that will give you the highest dose. So swallowing water is the easiest way of exposing yourself to toxins. And that's one of the main things we have to be concerned about with pets and livestock. I don't know about your dog, but my dog loves to swim, and she just drinks the water constantly while she's there. So they don't have a way of, telling themselves to say like, it's icky, I shouldn't drink that. I mean, dogs like stinky things. So it isn't necessarily going to be like this picture, a situation where the dog would say, I'd rather not, where I think most humans would say, I'd rather not. But, if you have a situation where the presence of cyanobacteria is there, but not to a bloom level, you still can have significant toxins present in that system. So you tend to get symptoms if you're swimming in sort of your mucosa, right? Your skin, your eyes, your nose, and throat and lungs, when you start to maybe have coughing or itchy throat. And then if you are consuming the contaminants, then you often have more symptoms in your gastrointestinal tract, or maybe in your head. You start to have headaches or dizziness, vomiting and diarrhea are some of the most common ones. And of course, that makes it complicated because a lot of different things can cause these symptoms. So you have to really be vigilant about your exposure and trying to think about, if you're experiencing any symptoms. Some of these symptoms can come on quickly, and some of the lingering health concerns, those take some time to develop the sort of stronger impacts. So there's a paper that came out in 2019 that talks all about the human health effect linkages to cyanotoxins. And it's staggering, really. All these different body systems that can be affected by cyanotoxins. And then from here, they've been implicated, but not proven to be factors in multiple different human diseases. The one that's getting the most research these days is ALS, Lou Gehrig's disease. There's some evidence that cyanobacteria toxins could be involved in that degenerative condition. So again, there's lots of research that still needs to be done in terms of the particular disease presentations. But these are body systems that are, central to our health and functioning. So it's really kind of something that I never really thought of before I started getting involved in this project. I'm a recreational swimmer and paddler, and I never really thought that I could be dosing myself with toxins just by doing healthy outdoor activities. So I don't mean by any means for you to shy away from doing those activities, but it's great that you all are here tonight to hear about, some of these things to just be aware of out there. So the adage goes, when in doubt, stay out. If you suspect a bloom, we'll talk about how to report a bloom in a minute. Stay out and keep your other animals out of the water. And if you can't, if you have touched the water, make sure to wash your body, your hands. And if it might be time to give Fluffy a bath if they've been in the green scum. And if your dog or pet starts to have some of those same symptoms, vomiting, shaking, difficulty breathing, extreme lethargy, it's good to call the vet. Dogs have shown some of the most severe cyanobacteria symptoms. There's been a number of reports of dogs dying, predominantly in the South, not so much in the Northeast, hours after swimming in contaminated water bodies. So there's sincere cause for concern, and it had to have been very traumatic. So we definitely want to try to prevent as many of those instances from happening as possible. And that's why we're starting to develop monitoring programs aimed at tracking cyanobacteria in local ponds. So now I'm going to tell you about work that I've been doing at the Harris Center on this front. So since 2019, the Harris Center has been collaborating with another local partner, the Norway Pond Commission, to sample Norway Pond according to this protocol set out by the Environmental Protection Agency, that's called the Cyanobacteria Monitoring Collaborative. And we follow these particular protocols so that they're standardized and allow comparison across water bodies. So we're halfway through the sampling season this year. Twice monthly sampling results in about 10 sampling dates per season. And so far, we're doing this at Norway Pond, but there's potential to expand this program at the Harris Center and beyond. And I think a long-term potential goal is to think about, can we get lake associations, trained in this process? Really, anybody can sort of go through the training to be part of this Cyanobacteria Monitoring Collaborative. And another great thing is that the EPA has set up a variety of tiers for sort of, how deep do you want to go in terms of monitoring the cyanobacteria? So there's sort of entry-level tiers and there's higher level tiers. And here at the Harris Center, we're doing all 3 of the tiers, which involves, looking for blooms, sampling the phytoplankton from these water bodies, and also analyzing the pigments that are found in the samples using a fluorometer device. And I'll talk about that here. So, these are like the days where it's like, pinch me, I'm at work, right? So here's my rig about to go out on the pond at Norway Pond. So if you've ever been to Hancock or you live in Hancock, you probably know this scene pretty well, how lucky we are to have this sort of jewel at the center of this town that I'm lucky enough to work in. So I'm out there every other week, more frequently if there's blooms reported by community members. As you'll hear later, all of us, everyone is an important observer in our efforts to increase water quality. So when I pack my gear, what do I have in my boat? We'll have a temperature probe, and I keep track of the temperature of the water at the surface and also the air temperature. I have an integrated tube sampler, which is basically little flexible tube with a weight on the bottom. And this tube is 3 meters long and it collects a whole water sample. So it's weighted, so it goes down in the water column, and then it has a string, and you pull it back up, and you basically get this 3 meter long, 1 inch wide whole water sample of the lake. And then every month I take, this is a strange looking fish that I caught here. This is a plankton net. And this allows me to collect sunshine in a bag, right? So this is going to go down to 3 meters as well. And we'll collect phytoplankton that's present in the water column. So then I use these samples to undergo a variety of analysis. We'll talk about that in a minute. So Norway Pond, such a great little pond. It's a shallow pond, maximum depth 20 feet. So this is what we call a mesotrophic lake. It's a warm water fishery. It doesn't stay cold because, and in the summertime, there comes to be a point where there's almost little to no thermocline. You just have warm water straight to the bottom. And because it's so shallow, light penetrates all the way to the bottom. So you actually can get photosynthetic autotrophic activity by cyanos and algae all throughout the depth of that water column. So quite amazing. So the Norway Pond Commission, thanks to Tom Chevenel especially, who every season puts out a sampling buoy at the deep spot. So I know where to go. So I always take the samples from the same spot. And then after I take my sunshine in a bag, my plankton net sample, I put it in one of these dark bottles. So that's one of the parts of the methods that I just love, is just like doing all this stuff on a boat in the middle of a lake. And this is before you even come back to the lab and start to do any of the analysis. So fluorometry, this uses those whole lake water tube samples that I talked about. And a fluorometer is going to evaluate the emission spectra of the two pigments that are most prevalent in the sample that are implicated in photosynthesis. I'm sure you've heard of chlorophyll before. Chlorophyll is the main photosynthetic pigment used by plants and algae. Cyanobacteria have chlorophyll as well, but they also have another pigment called phycocyanin that isn't exclusive to the cyanobacteria, but it's most commonly found within that group of organisms. It's popped up in a few places on that phylogenetic tree, but it's sort of housed significantly within the cyanobacteria. So the thinking goes is if you're looking for phycocyanin, you can use that as an indicator of the amount of cyanobacteria that you have in the system. Okay, so on the left side here, we see sort of pigments absorb light at a certain wavelength, and that excites those pigments, and they emit light at a different wavelength. And that excitation or emission light is what the fluorometer tracks. So it's this little small handheld device that's really straightforward. You use these small cuvettes, and you have sort of standards, and then also you then can do your lake samples. So here's what some results look like. All right, so fluorometry, the goal is that it can clue us into potential bloom activity. It isn't on its own a diagnostic feature, but it allows us to track some of the potential activity. So for example, here we see the levels of phycocyanin in blue and chlorophyll in green across the summer of 2020 at Norway Pond. And you can see chlorophyll levels were relatively stable. Phycocyanin, dramatic change starting in July. And what's interesting is the spike in phycocyanin was indicating the conditions for the bloom to set up. And when we sort of started to see this, the phycocyanin rising, we started to know, okay, there's something happening in this pond. But you really need to combine this type of analysis with scoping. So that's what we call cyanoscope. For these, we use the samples collected in the plankton net. We use this device called a zapper to separate the phytoplankton from the zooplankton. The zooplankton are the animals that show up in the net. Interestingly, the animals will float down here. We'll actually seek out this clear part of the tube, and the cyanobacteria stay at the top. So you can actually pipette off the cyanobacteria fraction. You get a little bit of algae in there too, and then you pop it under the compound scope and see what you get. And this all came in handy in August 2020 when a number of reports came in, including from Susie Spickle, who said, there's something happening in the pond. And I got out there on my boat and sure enough, saw these things, green, electric greenish blue tendrils. This is the Cyanobacteria Blob sort of stuck on a aquatic vegetation stalk. So they sort of, this particular type of cyanobacteria is benthic. So this organism actually started from below and bubbled and bloomed towards the surface. Underneath the scope, it looks like this. And this is what allowed us to identify the particular genera of the bloom at Norway last summer. And it turned out to be Oscillatory, which is a genera of cyanobacteria that's implicated in some toxin production. So the levels weren't high enough to trigger a beach advisory, but there was an alert issued. And signs were posted, alerts went out to try to discourage people from recreating in the pond. Here's another sort of microscope, cyanoscope sample from Norway Pond. This is a microcystis or a Phanacapsa. It's kind of hard to tell from this photo, but you know, these are small organisms in and of themselves, but when you get a preponderance of them, they can, have quite a different look. The Nubanusset Lake cyanobloom happened in November, even later than the Norway bloom. Usually, August, September are considered high bloom times, and blooms sort of typically subside often or die back by the fall, but we saw Dolichospermum, the first known bloom, first reported bloom on Nubanusset. So here's what an alert looks like when it comes from the state, the DES. This was Dolichospermum, formerly known as Anabina, a really common cyanobacteria species or genera in our area. All right, so let's talk about what you can do. Reporting a suspected bloom is easy, and environmental personnel can't be everywhere at once. So here's what you should do. If you suspect a bloom, write down lots of information, and most importantly, take photos, maybe more photos than you think you need to take, because they often can reveal different attributes. For example, location. Blooms don't always happen out throughout a whole lake. They can just be in one cove or in one area, or maybe just the beach area. So the date and time and weather conditions, and then you have two options. You can download this free app that's called Bloom Watch, or you can text them. If you're in New Hampshire, you can text them to your state agency at this number. And Amanda McQuaid is typically the responder here in New Hampshire, and she's awesome. So you don't need to take a physical sample. DES will follow up. If you want to get a little ambitious, though, you could try the stick test. And the stick test is you just take a stick and you stick it in the green globby. And if it sticks to the stick, it's likely green algae. If it looks like this, like hairy, kind of filamentous, it's most likely green algae. If it doesn't stick, it's more likely to be predominantly cyanobacteria. So that's a good trick. And when in doubt, report it, because it takes a while to really get a handle on what they look like. For example, here's some bubbly Mugiodia algae. This is a bloom, but not a cyanobacteria. It would stick to the stick. So that can be a good sort of entryway test for you. And then once you submit your observations, there's public beach protection programs that help respond to these potential threats. We will only see advisories from the state of New Hampshire if cells get above 70,000 cells per milliliter. So the state does the enumeration and counting, and then that leads to a variety of triggered advisories. Okay, so you can check some of those advisories. If you're here in New Hampshire, in the interest of time, I'll show you this quickly. This is what a beach advisory looks like in New Hampshire. You could always check it before you head out. If there are areas with the square with the exclamation point, those are cyanobacteria advisories. You can click on it and get more information. So if you're headed out to recreate somewhere, it's never a bad idea to sort of check water quality conditions before you go. It's updated every day in the summer and the swim season. Report blooms using some of those strategies we talked about. Don't swim. Use the Bloom Watch app if you're tech savvy. And if you reside near a lake, you might consider testing your septic system for leaks. New Hampshire Lakes and a variety of nonprofit agencies have a group of service providers that are sort of considered lake smart, so that they use practices that are conducive to water quality. Think about opting out or scaling back fertilizer use, especially during the bloom season, because of the leaching and the flushing of nutrients that we talked about. And one of the big things we all need to think about now is stop global warming. Think about what you can do as mitigation tactics, because we all play our role in the increase of Earth's temperature, and that's one of the key drivers. So driving less, switching to, renewable energy where possible, all those things add up. Dietary shifts, they are not, maybe direct influences, but really pivotal indirect influences on the system. And here's a bunch of references that I use. There are so many out there. So if you're interested in particular resources or know of good ones that you think I might be interested in, please send them my way. I'm really just starting to just get more and more involved in this topic with each passing month. I want to offer some thanks, especially to y'all for listening. My Harris Center colleagues and board members, you guys are the best. Amanda McQuaid and Jim Haney have really helped get my microbial feet under me. So thanks to them for all the training. And a number of EPA folks have offered standards and method support. And there's some local people that have really helped me. Norway Pond Commission, Tom Chevenel, H. Marie Lawrence took a lot of these microscope pictures, and she's taught me so much. And often I'm lucky enough to be out there when the VLAP, Volunteer Lake Association program is taking place, and Dick Warner is such a wealth of information about fresh water. So thanks to all them. And I'm happy to take your questions. I'll try to answer them as best I can. So thanks so much.
Karen, wow, thank you so much. That was fabulous, so informative and so much to think about. And yeah, just really, really well done. There are a few questions. One of them is, this is from Anne. She's curious about the Bloom Watch app, if it includes info on existing blooms.
Not as much. I would say there, if you want information on existing blooms wider than New Hampshire, then NALMS, the North American Lake Management Group, NALMS.org, I'll write it in the chat. They're the best for live stream, if you will, bloom information that's sort of wider than a state level. And then otherwise, you want to look at state level agencies. So New Hampshire DES. I don't know what state you live in, Anne, but every state will have a different sort of advisory web page. So like a lot of things, they're often not all in one spot. I would say Bloom Watch is best for reporting the bloom. And in that case, it goes to EPA, and then EPA contacts the state. Here in New Hampshire, Amanda is so amazing at what she does that I just directly contact her. And I find that that tends to get the fastest response because you sort of like cut out the middle group. And then often Amanda is then reporting that information to EPA. So it will get there either way. So that you have a couple different choices. And in theory, they should all produce the same effects.
Great, thank you, Karen. That was great. And Anne lives in New Hampshire, so she has the DES contact information too, which is great. There was a question about saltwater toxins like the red tide. If you could talk a little bit about that as well, are they related?
Sure, yeah, the red tide, it's always interested me because, we are Penn State. I, grew up going to all these Penn State games, and it always is amazing to me how University of Alabama has a microbe as a mascot, right? The red tide is their mascot, roll tide, right? Red tide is actually a dinoflagellate. It's an algae, not a cyano. In saltwater systems, it's primarily the algae, the true algae, eukaryotic algae, that are causing toxins and blooms. And the cyanos reign supreme in freshwater systems. It's not to say you can't have them in either place, but it tends, so red tide's a dinoflagellate. So it's just a slightly different structure, and hence the name. It has a little flagella and they can move around. So red tide is especially problematic because of all the shellfish farms that are in the Gulf of Mexico, and shellfish by their nature and bivalves, they're filter feeders. So if they take in the toxin, we get that same bioaccumulation, biomagnification effects that we talked about earlier. So it can be concerning. And that's why there's a lot of efforts to monitor marine systems, especially near fisheries.
Great. Can you just give us a little update? Anne's wondering who Amanda is, Amanda McQuaid.
Oh yeah, Amanda McQuaid is, she works at New Hampshire DES. So she is the sort of chief algal bloom, cyanobloom contact for the state of New Hampshire. So she has a staff that works with her, but she is a mover and shaker in the world of cyanobacteria. And she is the one that's, if you encounter a bloom, you will be encountering an interaction with Amanda, and she's awesome at what she does.
Well, Karen, so are you. And we really appreciate you taking the time tonight to get us all caught up to speed on our cyanobacteria and how fascinating and really such great work. Especially, I'm going to give a shout out to Tom Chevenel, who really got this program off the ground for the Harris Center, and then for Karen and the volunteers who've been working with Karen to make this program happen. And it isn't happening in other places yet in New Hampshire, right? This is kind of like a pioneer program. So great work and so good information for us. And I think we'll call it a night. So good luck to everybody in swimming. Keep your eyes open on the ponds. And Karen, keep up the fabulous work. So informative and it's just so bright. So yay, thank you.
Thank you all for coming. Great to hear for your interest. Thank you so much.
Yes, good night, everybody. Bye.